Hey guys, today we are going to talk about Lac Operon. Operon is a system in which two or more genes are under control of a single operator. In the system, regulation is done by two methods. First is by repressor protein and second is by Cap-Cam complex proteins. This is the structure of Lac Operon. It contains Lac I gene, Lac Z gene, Lac Y gene and Lac A gene with their common operator, and both of these gene sets have their respective promoter and termination sequences. Lac Z gene codes for enzyme beta glycosidase, Lac Y gene for permease, and Lac A gene for transacetylase. Transacetylase believed to add acetyl group on enzyme beta glycosidase, but its actual function is not known. Permease allow rapid entry of lactose into the cell, while beta glycosidase converts lactose into glucose and galactose or into allolactose. Repressor protein inhibits the transcription of enzyme. During transcription, Lac I genes were transcribed first as they are upstream of the enzyme gene. The product of these genes, that is repressor protein, binds to the operator region of enzyme genes. Hence, RNA polymerase cannot bind and transcribe further. Therefore, these proteins are also called as repressor protein or inhibitor protein. But not 100% gene expression is halted. Some amount of enzyme can be produced because of leaky expressions. When high amount of lactose is present, small amount of beta glycosidase is converted into allolactose. So in presence of lactose, allolactose binds to the repressor protein, due to which the shape of repressor protein changes and it cannot bind to operator region. An enzyme gene expression occurs. Another method of regulation is by cap proteins. There is one more region present upstream of the promoter of enzyme genes, which is called as cap binding site. When lactose is present in high amount, when the lactose is present in high amount, the concentration of cyclic AMP in cell increases, which binds to cap protein and activate them. This activated complex binds to the cap binding site and recruits RNA polymerase on promoter of Lac enzyme genes, and hence there is increase in the production of enzyme. But even if small amount of glucose is present in cell, the concentration of CAMP decreases rapidly and the gene expression slows down. This is called as catabolite repression of glucose. Because of this, glucose is consumed before lactose.